Hello, and welcome to this Nature.com custom webcast titled Micropatterning on EM Grids, a strategy for improving cell cryo-ET workflow. My name is Jay Shankarpan, and I will be your moderator. Today's webcast is sponsored by Alvéole. We'll begin the webcast with a presentation from Dr Leeya Engel of Stanford University and then end with a Q&A session. To ask a question, just type it in where it says type your question here and then press submit at any point during the webcast and we'll answer them today. And now over to Leeya.
Thanks, Jason. In this talk, I'll describe how to micropattern electron microscopy grids, a strategy that has improved our workflow for cryo-electron tomography of cells. Further details on the results that I present can be found in our recently published paper, Extracellular Matrix Micropatterning Technology for Whole Cell Cryogenic Electron Microscopy Studies. I'll start out by providing the motivation for combining protein micropatterning with electron microscopy. I'll give a brief introduction to the rapidly developing field of cellular cryo-electron tomography. I'll describe in detail the maskless photopatterning process that we've used to micropattern substrates for cryo-ET called EM grids. Finally, we'll have a look at our micropatterned EM grids in action. Micropatterning of extracellular matrix proteins, or ECM, is an established in vitro cell culture technique used to control cell shape. In this technique, ECM islands of arbitrary geometry are deposited on a cell culture substrate. As cells spread across the adhesive islands, they form attachments to the ECM called focal adhesions. In the example on the left, Manuel Terry and co-authors show that cell polarity is governed by ECM geometry. The top panel shows an adhesive fibronectin crossbow pattern designed to be an anisotropic cell microenvironment. You can see in phase contrast that a single cell spread on the micropattern forms a fan-like shape, and in the panel below, showing images of fixed cells on the micropatterns, the average distributions of vinculin, actin, and chordactin can be seen. The chordactin was restricted to the curved adhesive micropatterned regions, while the actin stress fibers, seen in the central panel, are enriched at the suspended, non-adhesive edges of the cells. This polarization of the cell cortex, due to the shape of the ECM, was further propagated to the internal organization of the cells on the micropatterns, and that was seen in the positioning of the nucleus, centrosome, and Golgi apparatus. On the right, you can see live cell imaging from Sanjay Kumar's lab, showing cells spreading on rectangular micropatterns. In this captivating video, you can observe the actin stress fibers in white assembling in parallel to the leading edges of the cells. This recent publication explored the dynamics of stress fiber network organization during cell spreading, finding that the rate of spreading depends on the micropattern geometry and on the initial adhesive position of the cell. So micropatterning is increasingly being used to understand how the size and structure of the cell microenvironment regulate the cell architecture, mechanics, differentiation, polarity, and function. But what does this modification of the cell microenvironment mean for the mechanical interaction between the cell and the substrate? When traction force microscopy is combined with ECM micropatterning, a correlation between cell morphology and the regulation of force transmission can be achieved. With traction force microscopy, it's possible to quantitatively analyze traction stresses by measuring the displacement field of the cell substrate, which should have known elasticity, and calculating from this the traction forces that the cell is exerting on the substrate. This has become a standard tool for many labs working in mechanobiology. In the figure on the left is a paper by Patrick Oakes from Margaret Gardell's lab, showing single cells assembled on micropatterns of the same area but different aspect ratio, starting with the circular micropattern in the leftmost panel. From the traction maps below, you can see that in areas of higher local curvature, you have higher traction stresses. The adjacent graph shows another interesting finding from this paper. The strain energy of single cells spread on micropatterns of different areas scales with the size of the cell, independent of the number of focal adhesions. On the right is a paper from Beth Pruitt's lab, which looked at cell-cell forces in cell doublets assembled on rectangular ECM micropatterns. The ECM micropattern can be seen in the schematic as green, and the cell-cell forces inferred from the traction maps are indicated in brown or orange arrows. In the middle panel, you could see EK adherent marking the location of the cell-cell adhesion. And on the right, what you can see is that as the aspect ratio of the rectangles was increased from 1 to 1.7, so essentially as these cell pairs are stretched, the cell-cell forces across the junction were increased. However, it was unclear how these increased forces were being sustained. So while micropatterning together with traction force microscopy can help us relate spatial constraints to cell-cell and cell-substrate mechanics, the nanometer scale protein organization underlying mechanotransduction, changes in morphology and cell mechanics, has not been observed. In other words, while we can use ECM micropatterning to regulate the strain energy and distribution of traction stresses in a cell, we don't know how cells sustain these states. Micropatterning on EM grids can be an enabling technology for super-resolution mechanobiology studies, as cryo-ET is the highest resolution tool available to us today for structural analysis of macromolecular organization inside cells. By micropatterning EM grids, we reasoned that we could enable direct observation of mesoscale organization in cells under morphological modulation, and that's what provided the motivation for this work. So, what are the steps that are required for performing cryo-ET on cells? First, you grow the cells on EM grids. Second, you vitrify the samples to maintain the native environment of the cell. We plunge freeze our grids in liquid ethane, cooled to minus 180 degrees Celsius, to create vitreous as opposed to crystalline ice. In cryo-EM, ice crystals are actually considered a form of contamination, and while you're imaging, these will come across as dark gray or black blobs. The cooling is performed in liquid ethane so that the ice is formed more quickly than crystals can form. So this vitreous ice is actually amorphous. Because cryo-ET is an electron microscopy technique where the electrons have to pass through the entire sample on their way to the detector, the total thickness must be below 500 nanometers. So depending on the cell type you're looking at and the region of the cell you're looking at, thinning of the sample by cryo-FIB milling may be required. Once you have the sample that you want to be observing with cryo-EM, transmission electron microscopy is performed at a series of tilt angles to create a series of projections. So electrons pass through the sample, which is being moved, and then 3D views of the samples, called tomograms, can be constructed from these different projections. So let's talk a bit more about these cryo-EM substrates called EM grids. The EM grids that we and other researchers use as cell culture substrates for mammalian cells for cryo-ET are comprised of a thin metal mesh, 3.05 millimeters in diameter, overlaid with a perforated carbon film called quantifoil. For cell culture, we use gold grids, which are less cytotoxic than copper ones. In our initial micropatterning work, we used grids that had 5 micron holes spaced at 20 microns from one another. More recently, for cryo-ET of cells, we've been micropatterning on grids with 2 micron size holes spaced 2 microns apart, and this has been giving us good results. I should mention that there's a variety of grid types for transmission electron microscopy, but the ones I'm discussing here are fairly standard for cryo-ET of cells. So the carbon film is approximately 12 nanometers thick, and you can imagine that these substrates are quite fragile. The carbon film can rip, and the thin gold mesh can bend during handling. Micropatterning of EM grids using microcontact printing was performed by T et al. Here you can see a cell assembled on a circular ECM micropattern, and they used correlative light and electron tomography on these substrates to show that cellular chirality arises from self-organization of the actin cytoskeleton. At the far right, you can see a tomogram showing radial and transverse actin fibers at the cell edge. Microcontact printing requires contact between a stamp loaded with protein and the substrate. In our hands, and on the grids we discussed, this was a rather destructive technique. Due to the fragile nature of our EM grids, we were in the market for a micropatterning technique that would maintain their structural integrity. Maskless photopatterning turned out to be exactly the micropatterning technique we were looking for. The PRIMO system by Alvéole is based on light-induced molecular adsorption of proteins and does not require physical contact with the substrate. In our case, this provided a huge advantage. In this setup, a UV laser is projected onto a substrate through the objective of an inverted microscope. A schematic of the setup can be seen at left, and a schematic of the light path inside the PRIMO module showing a digital micromirror array defining the pixels of the pattern to be projected can be seen at right. The system is controlled by dedicated software called Leonardo, which displays a preview of the digital pattern on the substrate, allowing for alignment of the micropattern to the substrate in real time. This is a huge benefit for micropatterning of EM grids. A specific photoinitiator, PLPP, enables the micropatterning of proteins with this system. Using a 20x objective, it's possible to achieve 1.2 micron resolution. So here you can see PRIMO systems in the lab. In this study, we used the setup at UC Santa Barbara on the left and the setup at Stanford on the right. Here, Leica DMi8 inverted microscopes have been outfitted with PRIMO, and you can see the optical case containing all the optical components behind the microscope and the power supply unit containing all the connections that power the optical case to the right. Without further ado, I will now go into detail on how we use these setups to micropattern EM grids. The schematic in A shows the ECM micropatterns in green deposited on the wholly carbon film of an EM grid and aligned to the grid so that the shapes are centered between the metal grid bars. In B, a cross-section shows the silicone stencil that we use to immobilize the grid carbon side up onto a glass surface for rinsing and patterning. This was very convenient, especially when I was new at handling grids, because once the grids were mounted on glass, I didn't need to worry about damaging them through handling. A photo of the grid with two edges constrained beneath such a stencil can be seen below in C. Once the grid was immobilized, we subjected it to atmospheric plasma at 30 watts for 10 seconds to render the surface hydrophilic. And you can see that pink gas in D1. We added polyalysine graphed polyethylene glycol at a concentration of 100 micrograms per milliliter to the surface and incubated for 1 hour to electrostatically adsorb the PLLG PEG to the surface. And this occurred via the positively charged PLL backbone. The densely packed PEG side chains provide the grid surface with anti-fouling properties against protein adsorption. We rinsed and added between 7 and 20 microliters of the PLPP photoinitiator to the grid. This is a pretty tricky step for beginners because it's very important not to let the surface dry out once the PLLG PEG has been applied. What I do is pre-fill one pipette with PLPP before pipetting off most but not all of the PBS from my sample using a different pipette in order to minimize the time that the surface isn't coated with liquid. Finally, we can load our glass bottom dish or glass slide containing the grid onto the stage of the microscope outfitted with PRIMO. The image files that we generated with ImageJ were loaded into the Leonardo software, and the samples were UV exposed at 2,500 millijoules per millimeter squared. With this exposure, micropatterning of each grid required between 30 and 40 minutes. Following exposure, we rinsed the grids and added the ECM protein of our choice, seen in green. In our case, this was usually gelatin or fibronectin at a concentration of 100 micrograms per mil. We incubate for 1 hour before rinsing again and then seeding our cells. The protein was selectively adsorbed to the exposed regions of the carbon film, and the cells, in pink, assembled on the micropatterns. Before we move on, I want to emphasize that I'm really happy to answer specific technical questions relating to this process following the presentation to ensure that you can easily replicate our results. I do think that maskless photopatterning is going to become a standard technique for people performing cryo-ET on cells and would like to help make it accessible. So with this approach, we were able to achieve large area micropatterns of arbitrary geometry at sufficiently high resolution. And in the image on the left, you can see green fluorescent protein tagged gelatin micropatterns positioned on intact carbon film. On the right, you can see rhodamine tagged fibronectin square micropatterns in red, overlaid with a bright field image of the 200 mesh quantifoil grid it was deposited on. The holes in the carbon film in light gray are 5 microns, and the square micropattern can be seen centered between the gold grid bars in black. So how do we achieve such alignment? As I mentioned a few slides ago, the Leonardo software allows you to observe a projection of your digital pattern on the substrate in real time. So while developing this process, I would manually rotate the grid to fix its orientation prior to creating an array of my digital pattern to fill a 12 by 12 region of the grid. More recently, Alvéole has come out with new software that will detect the orientation of your grid and perform alignment automatically. With the new dedicated software, the user can either repeat a single pattern across the entire surface of an EM grid, as shown here, or combine a series of different micropatterns onto the grid. And from my personal experience, I can tell you that this is a time saver. So how do the cells like these substrates? We showed in our paper that maskless photopatterned EM grids are viable cell culture substrates that promote cell growth and can confine cell spreading to within the boundary of an ECM micropattern. The figure on the right shows epithelial cells grown on and confined to square and circular fibronectin micropatterns of different sizes. This is an exciting result for reasons beyond the mechanobiology that motivated our study. It turns out that positioning cells on grids is extremely helpful for getting the most out of your grids when performing cryo-ET. We believe that maskless photopatterning can increase the success rate of cryo-ET data acquisition through positioning cells and specific areas of interest of cells at favorable imaging positions. Here's a figure from a recent preprint from Julia Mohammed's lab in collaboration with Manuel Thierry's lab, comparing cells plated on EM grids that were blanket-coated with fibronectin and EM grids with fibronectin micropatterns. The white arrowheads in the image on the left indicate that without micropatterning, only a small fraction of the cells is optimally positioned, as opposed to the case on the top right, where there is a higher yield of optimally positioned cells within the dotted yellow lines, indicating micropattern squares. In fact, and this is anecdotal, we and other researchers have found that cells in particular like to settle above the gold bars where they're inaccessible to the electron beam. I think that this may be due to the higher stiffness in those regions, but I haven't seen a study that went into this in detail. Depending on the cell type, cryo-FIB may be required for cryo-ET of cells. So here the researchers used HeLa cells. And so to keep the total sample thickness below 500 nanometers, they had to thin them. What you can see in E is yellow rectangles indicating the patterns for milling. And F shows the 90 nanometer lamella produced from the cell. And G shows a 6.8 nanometer thick tomographic slice of the nuclear periphery of the cell, where a coated vesicle, nuclear pore complex, polysomes, and microtubules can be identified. So the researchers showed that cryo-FIB milling is compatible with micropatterned EM grids and cryo-ET. It's possible that fluorescent micropatterns can even potentially be used to guide cryo-FIB milling of cells for cryo-ET. In our work, in addition to PTK1 cells, we tested a variety of cell types on micropatterned grids, MDCKs, HFFs, HUVECs, LECs, showing that this technique is quite versatile. Here, you can see live cell microscopy of fibroblasts assembling on rectangular fibronectin micropatterns over a half an hour. In the middle panel, you can see those same cells after being fixed and stained. The actin is in magenta, and the nuclei can be seen in blue. At the right is our digital micropattern, which dictated the shape of the ECM. Before settling on maskless photopatterning, we explored other micropatterning techniques for EM grids, such as microcontact printing and photoresist liftoff assisted micropatterning. One advantage of microcontact printing is that it's widely available and a low-cost technique. You literally stamp protein onto the surface of your choice. When we compare this technique to maskless photopatterning by printing rectangles on continuous carbon films, we saw that the features produced were not as sharp as with maskless photopatterning. Below you can see the average binarized fluorescent images of fluorescently tagged gelatin rectangles on continuous carbon films produced using these two techniques. The template is at right. When we microcontact printed on grids, we unfortunately ripped about 21 out of every 36 grid squares, which is a good deal of ripped carbon film. Because this process involves creating a physical stamp, it also does not allow for rapid prototyping, whereas when using virtual masks, the cost per design iteration becomes highly reduced. So even if you are very gentle and get microcontact printing to work for your grids, you don't really have a built-in method to align the micropatterns to the grid, as you would with maskless photopatterning. When it comes to photoresist liftoff assisted micropatterning, we were able to successfully perform the first phase, which is photolithography. So what you can see is alignment of these rectangular photoresist patterns to the center of the grid bars. In the photoresist liftoff assisted micropatterning technique, PLLG PEG would be deposited, and then the photoresist would be removed in an organic solvent called NMP. When we tried this on the grids and looked at them, we didn't see any carbon film following the immersion in NMP. We think that this process can potentially be compatible with micropatterning of EM grids, but it would have to be modified to employ a more suitable organic solvent. It's possible that other grid types could also work, but that would have to be explored. One final capability of PRIMO that would be impossible with the former micropatterning techniques is micropatterning of protein density gradients. In their advanced materials publication, Straley and co-authors showed that the density of adsorbed molecules scales almost linearly with the dose of UV light, and you can see that in C. In other words, by using local cleavage of PEG chains, or photoscission, we can tune the anti-fouling, making gradients a possibility. While we have not yet used PRIMO to pattern density gradients on grids, it can be an interesting direction for researchers interested in cell migration or polarization. So the image in A shows a digital pattern with varying gray levels, and in B, palm microscopy was used to quantify the fluorescence of the printed GFP pattern. In D, 3 different gradients were patterned on a substrate and imaged with epifluorescence microscopy. Logarithmic at the top, linear in the middle, and exponential at the bottom. The fluorescence intensity was measured along the dotted red lines of figure D, and the fluorescence intensity profile of each of the pattern gradients was plotted in red and E, showing good agreement with the theoretical expected profile in blue. To summarize, we have demonstrated a robust, versatile, maskless photopatterning technology for depositing ECM proteins on EM grids with programmed shape. This eliminates the need for physical contact with the EM grid surface for micropatterning, is compatible with a variety of cell types, and can be used to position cells at specific locations on the grid. We hope that this technology enables you and other researchers to structure in vitro cell microenvironments to correlate changes in nanometer scale organization at cell-cell and cell ECM contacts to strain energy states and traction stress distribution in the cell. And by reproducibly positioning your cells at specific locations on EM grids, we hope that you can improve your cryo-ET throughput. Thanks for listening, and I want to thank my postdoctoral advisors at Stanford, Beth Pruitt, Alex Dunn, and Bill Weiss for their guidance, and members of the Pruitt, Dunn, Weiss, Hanin, and Volkman labs who contributed to this work. Thanks to the team at Alvéole for all your help with PRIMO and developing the maskless photopatterning protocol. We're looking forward to using the new photopatterning materials that you've recently developed. And of course, thank you to the funding agencies that supported this work. And with that, I'm happy to take any questions.
Thank you for your presentation, Leeya. It's now time for the Q&A. So I'll ask Leeya a question, just type it in where it says type your question here and then press submit. Our first question, Leeya, and this one asks, when performing cell culture on micropatterned EM grids, are you limited by time? And this person also asks, does the surface coating or pattern expire at some point in time?
Yes, this is an important thing to keep in mind when you're working with these micropatterned EM grids. And so with the chemistry that I've presented here and that we've also published in our paper, you definitely want to plate cells within about 48 hours. We know that the passivation, so the PEG layer, should be stable for between 48 and 72 hours. But there are some new chemicals that are coming out, or new protocols that have been developed by Alvéole, where you can use, instead of, say, PLLG PEG, you can use a PEG SVA chemistry on top of polyol-lysine, and this should give you a more stable passivating layer. I don't expect the ECM protein to go bad. Mainly, the passivating properties outside the micropattern would probably expire at some point in time or degrade with time.
Excellent. Thanks, Leeya. Our next question asks, how many cells, so cell density, have you been plating?
Yeah, this is a really good question. It's going to depend heavily on the cell type. So, of course, different cells behave differently. For example, when I was working with fibroblasts, you can get the cells to fully spread in about 2.5 hours. Whereas when we were working with MDCK cells, you're actually going to want to leave them overnight to get them to spread across these micropatterns. So if you're looking to get about 1 cell per micropattern, what I will typically do is I'll make a somewhat dense cell suspension. And then what I'll do is add 5 microliters of this cell suspension to the grid, and then I'll take it to the microscope. Most people have a microscope in their cell culture room. I'll have a look. I'll change the focus up and down so they can see roughly how many cells are there. You want to be going for 1 or 2 cells per square. And if it doesn't look like enough, I'll add another 5 microliters of cell suspension. What I'll do after that, I think it's better to overseed a bit if you're unsure and just getting started. And then what I'll do, I won't fill the entire, say, MatTek dish with media. I'll put it in with this droplet of about 15 microliters of media and cell suspension in the incubator. Come back after 2 hours or so, once the spreading begins, and rinse off the non-adherent cells. If you don't rinse them off, then you'll get more cells attaching. And then I'll do that by adding about 2 microliters. Say it's a 35 millimeter mastic dish. I'll just fill with media and then leave it in the incubator overnight for cell types like MDCKs, so epithelial cells or endothelial cells, which take a little bit longer to spread. Yeah.
Excellent.
So it's
Thanks,
not an
Leeya.
exact science, I guess, is what I was trying to say, but try to eyeball it. Yeah.
Excellent. Our next question asks, have you tried using the micropatterns on any clinical samples?
Oh, that's such a great question. We're definitely moving toward that. As you go clinical, I think it's really important to keep sterility high. We have been using primary cells, primary neuronal cell types, but we're not far along enough to really have anything interesting to say about it. Stay tuned. There's no reason it shouldn't work. But for each cell type and for each ECM, there's a little bit of optimization that's going to be required for the process. So whether it's, your ECM incubation time or your cell incubation time or the shape of the micropattern, the density of the protein, etc. So we're working on something right now, but it's not there yet. Yeah.
Excellent. Thanks, Leeya. Our next question asks, from slide 8, it looks like both PRIMO systems used in this project are kept outside of a tissue culture hood. How do you maintain sterility of the grids that are micropatterned using maskless photopatterning?
Yeah, so this is especially important if you're working with a more finicky cell type, of course, or if you're trying to perform a longer cell culture. So for, as I mentioned earlier, with fibroblasts, you can get away with a pretty short cell culture time or incubation time where sterility is going to be less of an issue. But if you want to do something a little bit more complicated, say keep cells alive on micropatterns for a week at a time, sterility starts to be a lot more important. What we've done is, you can take your grid and place it in the bottom of a glass mastic dish and secure it in place. Close the mastic dish inside of a biohood, and then you can even close it with parafilm to ensure that no air is getting in from the outside, and then place that on the stage of the PRIMO and do all of your rinse steps in the biohood. But again, I think it really depends on the kind of cell you're working with and what you're trying to get at, because for a shorter incubation time, you can actually probably get away with doing your rinse steps right next to your PRIMO system. I think sterility is more of an issue in the longer term cell cultures. Yeah.
Brilliant. Thanks, Leeya. Our next question asks, have you tested any micropatterns to generate single cell to cell contacts on EM grids?
I'm not sure I understand what this means. Does it mean to optimize for single cells or for cell doublets? Let me see. So if you're trying to optimize to get single cells or cell doublets, what I would do is I would look at the literature to find micropatterns that have been transferred to gels or that have been micropatterned on glass, and then the cells have been examined on those. So, for example, Chris Chen has a really nice paper on endothelial cell-cell junctions where he patterns these bow tie patterns, and then you have, so it's basically two triangles meeting at their points, and there you're going to probably get a higher number of single cells that assemble one on each triangle and then meet at the point where the triangles meet. So there's a lot to be found in the literature about optimizing the micropattern for the kind of question that you're interested in. And so what I would do is I would look at at mechanobiology literature, and then, which is also really interesting because you have the traction force microscopy to give you a sense of what's going on with the cells in a big picture sort of way, and then adapt those to your grid. Yeah.
Excellent. Thanks, Leeya. Our next question asks, can the patterns be precise enough for neuronal projections?
So yes, and some of us at Stanford, there's at least 2 groups who are actually working on this right now. So yeah, time will tell, I guess. But yes, we are trying. I mean, I haven't tried anything submicron, but micron scale micropatterns should be able to give us some interesting insight, or at least some helpful positioning for neuronal cell culture.
Excellent. Thanks, Leeya. And our next question asks, is it possible to sort of scale up for larger scale tissue interactions, for example, like embryonic bodies?
Ooh. Well, for starters, I think that might be a bit tricky to observe with cryo-electron tomography because they're a bit thicker. So I guess you would almost definitely need some cryo-FIB spinning. Could you position them with PRIMO? I would say it's likely, but I haven't tried it. That would be kind of interesting if the person who wrote the question wants to kind of email me offline. It could be an interesting thing to think about. I'm not sure what ECM protein you would use, but if they stick to some ECM protein, there's no reason why it wouldn't work. Just passivate the surface where you don't want them to go and hope that they settle in the other regions. I mean, if it's a roundish body, I would just micropattern filled in circles and hope that they settle on those ECM regions. Yeah.
Excellent. Yeah, definitely sounds like one to follow up after the webinar. And just a quick reminder to our live audience, you still have time to ask Leeya a question. To do so, all you need to do is type it in where it says type your question here and then press submit. Our next question, Leeya, asks, does the deposited ECM protein on your grids bridge the holes in the holy carbon?
So from our imaging, it looks like the protein, there is no fluorescent protein in the holes of the holy carbon, but the cells will bridge the holes. So when you're trying to do your electron microscopy, you can send the electron beam through the hole and image the cell, but we don't see any protein in those holes, no.
Excellent. Thanks, Leeya. Our next question asks, if using this technique with protein samples, do you expect the proteins concentrated on the carbon film instead of inside the holes?
Oh, that's basically the same question. I guess these guys were thinking along similar lines. So in our experience, and maybe actually I'll add a little bit of information here, because if you are interested in getting your protein in the holes, what you could consider doing is decreasing your blotting time. So I'll add a little bit of background in case that didn't make sense to some of the people who aren't doing cryo-ET. So you take your holy grid, you load it into a tool for plunge freezing, and you blot off the excess liquid. So what we've been using for the grids that we've been using, so these 2 micron holes spaced 2 microns apart, 200 mesh grids, I usually use about a 1.5 second blot from the gold side, so only one side. And so I guess theoretically, if you did want for there to be protein in the holes, and the protein is what you were interested in, and not cells that have spread out on a protein micropattern, what you could consider doing is micropatterning your grid using the PRIMO system, and then applying your protein with a short incubation step, and then instead of rinsing it, blot from the other side using 1 second or even a half second and see if that works. It could be interesting. I have not tried it because I'm more interested in things at the going on inside of the cells. Yeah.
Excellent. Thanks, Leeya. Our next question asks, you mentioned that maskless photopatterning is compatible with different cell types, but how compatible is it with EM grids made of different materials?
Yeah, so in this talk we actually showed, we touched on works that use different kinds of grids than we use. So we've been using these, I'll just remind the audience, we've been using these holy carbon films on gold. And we've also seen a paper which used silicon dioxide holy films on gold. And then I didn't go into the grid type in that early paper that we mentioned where they did combine cryo-ET with grid micropatterning using microcontact printing, but it was actually quite different from the ones that we use. And so I would expect for the process to be compatible with different materials. You may need to change certain parameters, such as the time in plasma. So for silicon dioxide, I might actually go a bit higher than 10 seconds, although I haven't tried it. What we've presented in our paper is the way that we micropattern the holy carbon coated grids. But I do expect it to be compatible with other grids with small, with a little bit of tweaking of the parameters, maybe even the incubation time in the ECM protein. Yeah.
Excellent. Thanks, Leeya. Our next question asks whether you can talk a little bit more about the silicone stencils you use to immobilize the grids. Are these harmful to the carbon film?
Okay, so we have actually, in some cases, started to move away from using these silicone stencils. As I've gotten better at handling the grids, I feel more confident in my ability to do that. And so it turns out if you're careful, and I'll start off by I guess quite literally answering the question. In some points, if you add the stencil to the grid and then pull it off, it can take some of the carbon film with it. So so we would prefer to keep the carbon film intact, but of course this is a function of how you placed the stencil on the grid. So it should really only be touching the very edges of the carbon on the grid. So it's not such a big deal if it pulls off a tiny bit. You still have the rest of the grid. But what we've started doing is actually making a droplet of the PLPP, of the photoinitiator, within a silicone stencil, which is slightly larger, so a 4 millimeter size circle, and then inserting the grid into that. And so if you are worried about the film integrity, if you're seeing that your grids aren't surviving the plunge freezing and you think that this has to do with the fact that you're pulling carbon off when you remove the sticker or the silicone stencil, then you can actually try this alternative method. You just need to be a little bit more careful. Yeah.
Excellent. Thanks, Leeya. Our next question starts off by saying that this individual works with cells that are able to pull out of the fibronectin from the substrate if the line width is around sort of 1 to 2 micrometers. And what they've been doing is they've needed to form some sort of covalently bound fibronectin to the substrate. So their question is basically asking, did you also proceed to covalently bind on the grid?
Well, what we've done here is we have this polylysine graphed polyethylene glycol layer where we use the photoinitiator to degrade the PEG. And so what the, in this case, fibronectin, right, what the ECM protein would be coming in contact with is probably the polyL lysine. So it should bind pretty strongly, but I think that you would have to try it out. So what I would recommend doing is to do a little bit of live cell imaging if you can, to just make sure that, you know, I don't know exactly how long this individual performed cell culture on the fibroblast for. I know that when I did this with fibronectin using this PLLG-PEG chemistry with the photoinitiator, I didn't see the cells coming off. They actually spread quite nicely on the rectangles. I think that was in slide, yeah, I had some live cell microscopy of this in slide 14. Yeah, so I definitely didn't see any, I didn't see the cells pulling out the fibronectin. I saw the cells spreading on the fibronectin micropatterns, and you can pretty clearly see that they adopt this rectangular pattern. So my guess is that it would work, but you know, I would try it before going to use your electron microscope. I would try to perform some live cell light microscopy on the samples just to make sure that it works for you, you know, if you're doing a longer cell culture than I did. Yeah.
Perfect. Thanks, Leeya. And our next question asks, do you have any other tips for researchers who want to use micro-patterned EM grids for cryo-ET?
Let's see. Well, one thing that we've started to do, if you're using gold fiducial markers, so we've found that it's helpful to mix those gold fiducial markers in with your ECM protein, and following the UV exposure, if you can incubate with a mixture of ECM protein with gold fiducial markers, then when you're trying to reconstruct your tomograms, you have fiducial markers both below the cells, so on the ECM that they're going to be attaching to, and also above the cells, because you would be adding some during or right before blotting and plunge freezing. So that's one thing that we've been doing. Another tip, I would say plan your time really well, because you want to biopassivate the surface as close as you can to doing doing the actual exposure with PRIMO, and then you also have to make sure that you have cells in culture ready to plate following your ECM incubation. So I would say to really take the time to sit down and plan your week, because micropatterning the EM grids, it does add, I would say, between a day and a day and a half of work, depending on how many grids you want to micropattern. I'll usually do about 4 at a time. And then when I have my actual session on the microscope, I probably will only get through about 2 of them, because there's just so many cells that are optimally positioned that you can get a lot out of a grid. But 4, it takes a while, because if you think about it, there are a number of incubation steps that are an hour each. There are a number of rinses, and that's not even including, you know, the exposure time. And so you also need to make sure that your cells are going to be ready to split right when you're done with that. Yeah, so those, I guess, would be my sort of tips for anybody starting to use this process. And you know, especially if you're using this process for the first time, I would even suggest that you do your biopassivating on a separate day from your UV exposure, because otherwise it can make for a very long day if you have to check back on the protocol every step. I typically do them all in the same day and then plate my cells in the evening and plunge freeze the following morning, but I would start out slow. So those are my tips.
Excellent. Thanks, Leeya. Our next question asks, in your presentation slides, it looks like the pattern that appears, it looks as though the pattern appears to restrict the growth of the cell in a certain hollow ring or square. This individual was asking whether the red signal in the figure represents where the ECM is. Perhaps you can try to clarify things.
Yes, yeah, that's true. So what we found, and you see this in a number of papers actually, where a sort of frame-like pattern is created, and then the cell grows on this frame. Let's see, actually the two papers that were in slide 3. Oh, well, the latest paper that I mentioned in slide 3 from Sanjay Kumar's lab, those are actually these sort of frame-like micropatterns, as well as the paper from the Pruitt lab on the cell-cell contacts. You have these square and rectangular frames with a width of 8 microns. So the entire micropattern would be, say, 60 by 35 microns, but it's actually the area or the shape is defined by this 8 micron wide frame. And what I found for MDCK cells, at least, is that if you have a micropattern that's completely filled in, they can choose not to fill the entire micropattern. Different cell types won't do this. Endothelial cells and fibroblasts, I find, will spread across sort of a fully opaque micropattern. But what the cells are going to do when you have this sort of frame is they're going to spread, creating focal adhesions as they go along the frame, and this will allow them to really adopt the shape that you're going for. In addition, if you're looking at something like cell-cell contacts, so in the case of the paper from the Pruitt lab, you have the cell junction actually suspended, and that can be convenient for decoupling different things that are going on in the cell. For example, cell ECM contacts from cell-cell contacts. So yes, the red signal, that red outline that you saw, that is where the ECM is. There is no protein in the center, and the cells will grow across the ECM-free area. So they're not going to have any focal adhesions in that region, but the focal adhesions around the perimeter are enough to keep the cell suspended. And then there is a paper, I think also from Chris Chen, where he limits the area of the micropattern and shows that cells are not viable below a certain size. And there are a number of papers comparing focal adhesion density to micropattern area, to all of these different things that are going on when you plate a cell on a micropattern. So you do need to keep all of these things in mind and then design your micropattern so that it answers your biological question. Yeah, so I hope I answered that question.
Excellent. Thanks, Leeya. Well, I'm afraid that is all we have time for today. Before you leave us, we would really appreciate if you could spare a few seconds to fill in a very quick survey on your experience today. I would now like to thank Dr. Leeya Engel for her presentation and for answering your questions. I would also like to thank the webcast sponsors Alvéole, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.